Okay, thank you. All right, so yes, as Zach had said, I'm continuing today on my with this webinar series in regard to introducing peak 11. Today's topic is probably quite familiar to many. Basically, I will talk about the challenges of the identifications and quantifications of modified peptides. And along these lines, I will point out some of the improvements in sensitivity, as well as demonstrate Peak's quantification tools specifically applied to post-translational modifications. In terms of quantification, I will give a brief overview of our labeled methods. And lastly, I really want to focus today on a specific application using a triple SILAC approach in phosphoproteomics. Now, a bit of a background. We all know that the proteome is vastly more complex than the genome. So I have an example here of the human genome, which comprises about 20 to 25,000 genes, whereas the total number of proteins in the human proteome is estimated to over 1 million proteins. So what happens here is that firstly, we have one gene encoding multiple proteins. And then in addition, we have post-translational modifications, which can occur really at any step in the life cycle of a protein or in response to stimuli, which then exponentially increases this complexity in our proteome. And this complexity then requires state-of-the-art technologies, from sample preparation to data acquisition to data analysis to deal with such data. Today, there are more than 400 reported characterized PTMs. And these modifications happen as crucial molecular mechanisms that regulate diverse cellular processes, and the disruption in PTMs can lead to a dysfunction of vital biological processes and hence to various diseases. So clearly being able to accurately identify these different and quantify these different peptides, modified peptides, is of interest to many researchers. So here what I'm showing is a discovery timeline for really the very few, but some of the, I guess, major or most common, most abundant PTMs. You can see that it all kind of starts in early 1900s when we have a characterization of phosphorylation, and then right into 2011, when most of these major modifications are discovered and characterized. And this is really just in time for when the proteomics technologies really start taking off. Now, specifically in proteomics of post-translational modifications, we have additional challenges. Without going into really much detail, from the experimental point of view, those who have done PTM enrichments, purifications, well know that the current sample preparation techniques are really, really laborious and time-consuming. So in addition to the complex sample that we're dealing with, we can also have low abundances, low stoichiometries, or simply low yields from these enrichment procedures, or we can actually have the contamination often introduced, especially with the antibody or beads used within these sample preparations. So all of this together really results in a need for, on one end, highly sensitive instrumentation, LC-MS, but also highly sensitive, powerful, and comprehensive software tools, which can then deal with these kinds of data. Now, what and how Peak's approaches the identification of PTMs is, again, likely familiar. So we have sort of two different steps to the identification of the PTMs. One is through our PEAKS database search, where you would just select some of these major phosphorylations, perhaps most abundant or enriched in this case, and then we need to limit somewhat the maximum allowed number of variable PTMs per peptide. This is just a control for that search space. And then you get your PTM, the PEAKS database result. Now, another analysis that we also kept actually from previous versions is our PEAKS PTM search or our finder, which will, in addition to the database result, it will search basically all of the unmatched de novo sequences against either a selected list of modifications, so could contain these or additional ones, or really you can search against all of the built-in modifications, which is over 300 as per Unimod database. And then you'll again have your result displayed. So what this looks like, I'm just showing you here the PTM profile table from the summary tab from either PEAKS database or PEAKS PTM results, comparing the numbers of peptide spectrum matches from either of the searches, and you can clearly see that for each of these modifications I specifically selected here, we have the increase in the numbers of PEAKS PTM could find additional modifications, including the phosphorylation here, as well as, I believe I searched here the whole, all of the built-in modifications, so you get quite a few of these other modifications shown. And now another tool very convenient for looking at PTMs is our PTM profiling tool, which will produce a very nice visualization of really PTM stoichiometry in terms of the percentage of the modified peptides in your sample, in each sample or each replicate. This will be shown for all the confidently identified sites I've chosen here, the phosphorylation for a selected protein. And this is a rough estimation or calculation, but it can be useful, especially in phosphoproteomics and for, say, kinase reactions or similar. Now, what I want to point as new or emphasize new in PEAKS 11 is our deep learning boost, or the incorporation of the deep learning technology now for an improved sensitivity and coverage of the proteome. And if you've joined any of our previous webinars, you probably have learned that with the deep learning algorithm, we could achieve the increase in the identification rates, PSM rates of about 10 or more %. Now, to show you what this looks like in a specifically fossil proteome enriched data set. So this is a fairly large data set I use to basically search with the deep learning boost on and off, and just a quick comparison of these various different identified attributes that we show in our summary table. You can see that from no deep learning and to deep learning incorporated, we see right across these different attributes the increase in the number of the identified features and attributes. Now, just quickly back to that profile table where we look at only and specifically modifications, just to see if this deep learning is actually affecting the modified peptides as well. And again, I'm showing and comparing the numbers of PSMs here without and with the boost on, and you can clearly see that for each of these modifications, we see an increased number of identifications in our database search result. All right, so now that we've seen the improved identification rates, let's see how that reflects on quantification. And again, my colleagues in the previous webinars have mainly talked about label-free quantification, or LFQ, but today I want to focus on the label quantifications and demonstrate how PEAKS deals with such data, and then show you some visualizations that could be useful for you and the way the PEAKS-Q result will be displayed for you. So here's just a brief comparison of the label-free and label quantification, just a bit of a reminder. In label-free, we have our conditions to be compared, treated side by side, but still separately. So processed through sample preparation separately and acquired separately. Whereas in the labeled quantification, these conditions are mixed at some point during the sample preparation, and then all of the further processing and acquisition is really done on the one mixed sample. And this then results in, I would say, typically a higher quantitative proteome coverage for LFQ, however, a higher accuracy and reproducibility for the labeled quantification. Now, more specifically, we have typically less complex sample preparation with LFQ, less time consuming, with flexible study design, meaning really LFQ can be applied to many different organisms or different experimental setups. However, because we acquire these conditions separately, we require more instrument time with LFQ, which can be costly, and comparability of samples is difficult. With the labeled quantification, you will require less instrument time, will have easy comparability of samples. However, sample preparation can be more complex, with a bit of a sturdy, I would say, study design where it may not be applicable certainly to all organisms or all of your different experimental setups. And lastly, the reagents can be also costly. Now, amongst the labeled quantifications, I would say that metabolic labeling, such as SILAC, is deemed to offer the best accuracy. Here, mixing of conditions happens at the cell culture or cell lysate step, and then all of the further processing from protein precipitation to digestion, cleanups, enrichments to acquisition will equally really affect all of these conditions which are being compared. Now, with the chemical labeling, the conditions to be compared are mixed at the peptide level, which likely will result in more variation during the sample preparation. However, the main, I think, advantage of the chemical labeling is in its power, or the number of conditions that can be compared. Now, what we offer in PEAKS-Q is different built-in, both chemical and metabolic labeling methods. You can easily add your own. If you need help, again, let me know, let us know. We'll gladly help you to make it more specific for your own designs. In terms of the chemical labeling methods, I guess TMT would be probably most popular. We have up to TMT 11-plex built in with the purification factors. We're just working on adding the purification factors for the 16 and 18-plex, and hopefully that will be released soon. And then also different built-in SILAC labels. Again, if you have a need for different ones, please let us know and we will be happy to help you. So this is just a brief example of a chemical label. Here I have a TMT 16-plex
applied in a phosphoproteome analysis. So what this paper did, they used 4 different cell lines, so 4 different cell lines. Each was labeled with 4 TMT labels to basically fit into a TMT 16-plex labeling scheme. And what I wanted to see is how peaks will or could find and differentiate between these different cell lines in this complex data sets. And here's the result in terms of just a heat map. So different cell lines were grouped based on the by the TMT16, so 4, 4, 4, and 4. And I mean, you can clearly see this is the phosphoproteome, of course, and peaks could very, very well distinguish between these different cell lines in terms of their phosphoproteome. And just a slide showing the phosphopeptide level, because that's probably more something that you're interested in, the actual sites of modification and how these phosphopeptides change. So here you can actually either hover over a group profile to see these intensities and ratios, or you can simply look over a sample profile and even see really the reproducibility within each group and how well that was quantified. And then peaks will show you your ion match as well as your reporter ion with all these intensities normalized and raw intensities in terms of the comparison. And really the take-home message here is that peaks could quite successfully deal with this very complex data set and clearly differentiate between these different cell lines. Now, as I said in the beginning, I would really like to focus today in more detail on an application of a triple SILAC quantification in phosphoproteome analysis. Now, what I chose was the example, a recent example of a publication that used a chemical genetic approach in combination with a triple SILAC phosphoproteomics for deciphering of the substrates or targets of the CSMK2 or casein kinase 2 or CK2 enzyme. Now, why CK2? Well, it turns out that the levels of CK2 are upregulated in many of the cancer types, which then, of course, dysregulates other pathways, such as it delays apoptosis. And a lot of the work in this area is focused on the development of the inhibitor drugs for CK2 as a combination therapy for these cancer patients. Now, the way we designed the experiment, and I'm saying we because I was actually heavily involved in this project with my colleagues at Western University here in Canada. So the way we designed the experiment was we took our wild type, which was lightly labeled or unlabeled, and that was our DMSO control. Our medium label was our, again, wild type inhibitor drug treated. And then the heavy label was a chemically altered cell line, which was engineered to express what we call a triple mutant CK2 alpha, which has certain residues substituted to resist inhibitor drug binding and then basically keep the catalytic activity. So what we really hoped to achieve was to see certain level of phosphorylation in the light channel, so in our control. Then we were expecting the phosphorylation to be downregulated with the inhibitor drug treatment. And we were hoping to see the similar levels of phosphorylation in our drug-resistant cell line. This is what we were hoping to see the rescue of the phosphorylation.
So in other words, the aim of our study was to find the CK2-dependent phosphosites which were hypothesized to be inhibited in medium and rescued in the heavy inhibitor, CK2 inhibitor treatment. And I'm just showing you a raw spectrum triplet example where I see the light phosphopeptides at a certain level. You see the drop in phosphorylation for the medium, and then the nice rescue in our mutated cell line of this particular phosphopeptide. Setting this up in peaks is fairly straightforward. So I'm just going to go over our peaks-Q setup or workflow. Just go through the steps. First is the database search setup. This is a human cell line, of course. I added the phosphorylation STY as a variable modification. In SILAC experiments, you need to add all of the SILAC labels as variable modifications. Somewhat limit the maximum allowed number of variable PTMs. I upped this from about 3-4, we recommend to 5, just to kind of encounter for this complex sample. The deep learning boost was on, and for the peaks-Q, finally quantification part of the setup, I just selected the SILAC triplex built-in method, which is a very typical, typically used for triple SILAC, I guess, experiments with the lysine 4 and 8 and arginine 6 and 10. Being the isotopic labels. And then I just added all of the replicates in the same group, and you look at the different conditions of light to medium and heavy. I did have multiple time points, so I added each time point in a separate group. So here's just a quick look at the summary results. Some of the statistics, 180, almost 4,000 identified spectrum matches. From these identified peptides, you can actually easily, I typically check for this in my experiments, you can check for phosphor enrichment efficiency, but simplified, it's very simplified, but I calculated the ratio of the total number of phosphorylated peptides and total number of peptides. And in my experiment, it was at 98%. So it's just a quick, easy checkup of your perhaps sample preparation efficiency. And here's just statistics from the peaks-Q results. So quantified numbers of different quantified attributes. Next, I would like to show you some of the visualization tools that Peaks offers to help you analyze and understand your data. And one such visualization is ratio distribution histogram. In this particular experiment, we have again a phosphoproteome perturbance in the medium and heavy labels. We're hoping to see, of course, no downregulation in the heavy. Here I have the 4 and 24-hour time points shown. What Peaks does is it calculates the medium to light ratio and heavy to light ratio, where light is our control. So what I'm looking at here is the change in the medium channel, and at 4 hours you can see that there's already some downregulation happening in the phosphoproteome with this drug inhibition. At 24 hours you can see a lot more downregulation in phosphoproteome. Now looking at our drug resistant cell line, at 4 hours we see almost perfect kind of rescue of the phosphorylation, and then at 24 hours we start seeing some downregulation in our heavy medium or heavy label, and we're debating it. I mean, this is a prolonged exposure to drugs, could be other processes that are kicking in, or other kinases that are actually being downregulated, and that's what we're now seeing. Now, another nice way of showing and looking at your phosphoproteome perturbations is looking at the heat map, and I could easily spot in my heat map that I kind of cropped some of these phosphoproteins, which clearly are downregulated at 4 and 24 hours in the medium drug treated cell line. This is the 0 hour time point. This is the 4 and 24 medium. This is the heavy. So this is hopefully the resistant cell line. And it's fairly obvious that inhibition is in the medium channel. So very likely these phosphoproteins will contain some of the phosphosites which are CK2 dependent. And now you can either click on either of these different proteins, or what I did, I actually clicked on the whole cluster as done by the peaks to show you variation trend chart. So it'll be displayed right away for you, where you can additionally investigate and see which phosphoproteins are perhaps not as inhibited or affected by this drug, or which are more inhibited and then even more inhibited. So this is the medium channel again at 24 hours. We also can recognize some of the what we call fast responders, which are heavily inhibited and at 4 hour time point. What is really lovely to see in this particular for these proteins is a really nice rescue in the heavy 0, 4, 24 hour treatments. And still at the phosphoproteome level, I'm just looking at the proteins table. Again, peaks will have all these ratios for the medium for each group. So 0, 4, and 24. Nicely displayed in color, so you can easily spot these proteins again, which likely will contain these phosphopeptides or phosphosites, which are likely inhibited or CK2 dependent, again, phosphosites. And if you have a confidence site, it will actually display the position of that site within the protein. Now, what really the aim of this experiment was to find the CK2 dependent targets. And of course, we need to go down to the peptide level. So this is the peptide table. Now I'm looking at one of these phosphorylated peptides. Again, I can easily spot here the inhibition in the medium channel and a fairly nice rescue in the heavy channel. What this will show you is, well, MS2 for the selected scan or match, and then the MS1 here, it's a control, so we don't see the difference between light, medium, and heavy. Of course, the feature for each of these light, medium, heavy phosphopeptides as calculated by peaks, then the ratio or percent of the monoisotopic first and second isotope in each of the labels. And really what I like to look at is this extracted chromatograms for, again, the monoisotope, the first and second isotopic peaks for light, medium, and heavy. So light is a red, in red color, medium is in a orange, and heavy is in green. Now this is control, and what we see is about, the same levels of phosphorylation in this control sample. Now, if we go to a 4 hour time point, so 4 hours selected here, we immediately see, right, we see the drop in the phosphorylation for the medium channel, and really beautiful rescue, I would say, in this particular case by the mutated cell line. Now, if you go even further to the 24 hour time point, what we see now is, well, more downregulation in the medium channel, but also some downregulation in the heavy. And we're debating, it could be that this site is also regulated by perhaps not only CK2, but other kinases, which may be maybe some sort of off target and inhibited.
And now, while talking about these off targets, so just as important as it is to find your substrates, you want to see the off targets to kind of characterize the drug. And this is also quite easy in peaks because you can, again, follow the coloring scheme here. And I just picked a peptide, which I saw that was downregulated in the medium channel, but also downregulated in the heavy channel. And now I'm going to compare the control. So just a bit of a mismatch, but still about the same levels, light, medium, and heavy. Now, if you look at the treatment, I believe this was at 24 hours, you now see a really significant decrease in phosphorylation in the heavy channel. So this is a rescue condition which should not be inhibited. So clearly this is some sort of off target or something else is happening, and it should be likely further investigated. Now, with my example so far, I'm hoping I could demonstrate that different visualization tools in PXQ can be very helpful for elucidating, for understanding the phosphoproteomics data, and perhaps it can be helpful for your own experimental designs and analysis. Now here, the one question still remains, and that is how efficient is PEAKS in finding these CK2 targets? So in our manuscript originally, we used MaxQuant to quantify our phosphopeptides, and based on 1% PSM FDR and all valid areas cross-replicate, so this is quite strict. MaxQuant could find 848 or quantify 848 phosphopeptides. PEAKS, using the same filtering, same analysis, could find a few more of these quantified phosphopeptides, 927. Now, when it comes to the actual CK2 targets, as defined in terms of what we looked for was our ML to be inhibited 1.5-fold or more, and to see really no change in HL. So this was our kind of filtering. So the HL would be somewhere around 0, right? And then the inhibition in the ML. And CK2 targets identified by MaxQuant is 46. Now, using PEAKS and the same filtering, we could find 66 CK2 targets, so about 30% more. Now, I do need to say that when I looked at the overlap between these targets, there's about 50% of the targets found by MaxQuant are found also by PEAKS. So there's a bit of a mismatch, but I do have to say that the original drug that was used in this experiment was said to be by my biochemistry colleagues a dirty drug, and basically what happened when we looked at the whole phosphoproteome was heavily inhibited all across. So we know that this drug does not have the best specificity, and it does hit many other kinases but the CK2. So what I have actually, we got a hold of a higher specificity drug or a different drug that we wanted to test. And this is still a preliminary result. It is in submission, this publication, so I'm not going to go into too many details, but what I want to show you is how PEAKS could differentiate now between two drugs of different selectivities or specificities. So again, for PEAKS could find 66 targets for the lower specificity drug. For the higher specificity drug, it could find 153 CK2 targets. So over 100% more CK2 targets when we use a really good drug. And not only that, this drug is of higher specificity, but if you look, it actually has a higher potency and still very, very good and clear rescue. And the looking at the phosphoproteome, it is much less inhibited than with this drug.
Now, I'm not going to go into any more details about this. I'm going to conclude my talk with that slide to say that I'm hoping I could show you that PEAX-11 is efficient in analyzing post-translationally modified peptides, both qualitatively and quantitatively. Deep learning boost has improved the identification rates by more than 10%, and PEAX was successful in validating kinase targets using a triple SILAC-based quantification. And with this, I'd like to thank everyone for joining today and listening through my talk. And of course, if you have any questions, please go ahead.